12,000 feet up in the Himalayas, a snow leopard picks its way across a scree slope in air so thin that most predators could not sustain a full sprint for more than a few seconds. Its tail alone can measure nearly the length of its entire body, wrapped around itself for balance and warmth when the animal beds down for the night in temperatures that would kill most mammals its size. Its chest is barrel-shaped, built around unusually large lungs, and its blood is tuned to carry oxygen more efficiently than almost any other cat on the planet. Snow leopards evolved in isolation, high above the range of tigers or lions, on mountains far from any other big cat lineage. So when geneticists finally sequenced its full genome, they expected a clean, self-contained history shaped entirely by hundreds of thousands of years of solitary survival at elevation. What they found instead was a stretch of DNA that did not trace back to the snow leopard's own ancestors at all. It traced back to the shared ancestor of lions and leopards, two cats that have never lived anywhere near a mountain range like this one, left behind by a mating event that, according to everything scientists thought they knew about these species, should not have been possible. That single result was the first thread pulled on a much larger problem, one that eventually forced an entire field to rewrite how the cat family is actually built. Every living cat on Earth, from a lion weighing more than 200 kilograms to a black-footed cat small enough to fit in two hands, belongs to a single family called Felidae. Most classifications place the total at around 41 species, 40 wild cats spread across every continent except Australia and Antarctica, plus the one species living in millions of houses. The exact number shifts slightly depending on which authority is counting, since ongoing genetic work keeps splitting some populations once considered a single species into two, or occasionally merging populations the other direction. For most of the past 2 decades, the accepted picture of how those 41 species related to each other came from a landmark 2006 study published in the journal Science, led by geneticists Warren Johnson and Stephen O'Brien. Using DNA pulled from the autosomes, the X chromosome, the Y chromosome, and mitochondria of nearly every living cat species, the team built the most detailed feline family tree ever produced at the time. It organized every cat into 8 principal lineages, all descending from a common ancestor that lived less than 11 million years ago in Asia, spreading out across the world through at least 10 separate migrations over land bridges exposed by falling sea levels. The same study also exposed a separate problem entirely. Comparing the genetic dates against the known Felid fossil record showed that fossils underestimate when a lineage actually first appeared by an average of 76%, meaning the vast majority of the cat family's early history left no bones behind at all, only DNA. The tree itself, though, was clean. Each branch split once and never touched another branch again. One of those 8 branches eventually produced the cat sitting on a couch somewhere right now, though tracing exactly which wild population it descended from took another year of work. In 2007, a team led by geneticist Carlos Driscoll analyzed DNA from 979 cats, from pampered housecats to wild animals trapped as far away as Kazakhstan, and found that every domestic cat on the planet descends from a single wild subspecies, Felis sylvestris libica, the Near Eastern wildcat. Domestication began roughly 10,000 years ago in the Fertile Crescent, most likely when wildcats started following stored grain and the rodents drawn to it into the earliest farming villages. At least 5 separate wildcat founders from that region contributed to the modern domestic cat gene pool. At the time, it was treated as one of the cleanest single origin stories in the history of any domesticated animal. That same research group had already sequenced the domestic cat's own genome for the first time earlier that same year, using DNA from a 4-year-old Abyssinian named Cinnamon, whose pedigree traced back several generations to Sweden. The project identified roughly 20,285 genes and gave researchers, for the first time, a reference genome to compare every wildcat and every big cat against. One of the wildcat populations included in that original comparison, the Chinese desert cat, was later found through further genetic analysis to be different enough from the others to warrant full species status of its own, reclassified in a 2017 taxonomic revision as Felis beati rather than a subspecies of Felis sylvestris. Even the housecats family tree, treated as settled in 2007, kept getting redrawn as the genetic tools improved, the same pattern that would soon repeat itself on a much larger scale with the big cats. That clean, orderly picture held up for the smaller cats. It did not hold up for the biggest ones. 5 species make up the genus Panthera: the lion, the tiger, the jaguar, the leopard, and the snow leopard. Together they represent some of the most recognizable predators alive, yet for years genome sequencing had focused unevenly on them. The lion, the tiger, and the snow leopard already had fully sequenced genomes, largely because captive populations made high-quality samples easier to obtain, while wild, elusive species like the jaguar and the leopard lagged behind. If you're finding this interesting, be sure to subscribe to understand more DNA from the past. That first round of sequencing, published in 2013, produced the most direct comparison yet between a big cat and the animal asleep on a windowsill somewhere. Researchers sequenced a 9-year-old Amur tiger from a South Korean zoo alongside a white Bengal tiger, an African lion, a white African lion, and a snow leopard, then measured each genome against the domestic cat as a baseline. The tiger came back 95.6% identical to the housecat genome, despite the two lineages having split apart roughly 10.8 million years ago, essentially the same age as the root of the entire cat family tree. In other words, a tiger and the cat curled up on a couch are about as distantly related as any two members of the whole Felidae family can be, and they still share the overwhelming majority of their DNA. That same 2013 study pinned down one of the snow leopard's high-altitude adaptations to a specific gene, e.g. LN1, carrying a mutation that swaps one amino acid for another at a single point in the gene, a change tied directly to how the animal's body handles low oxygen at extreme elevation. It was at the time treated as a clean example of one species independently evolving its own solution to an extreme environment. In 2017, an international research team closed the Jaguar and Leopard gap, sequencing both genomes for the first time and comparing all 5 species in a single unified analysis, the most complete genomic picture the genus had ever received. Across the 5 genomes, researchers identified more than 13,000 genes shared by every living Panthera species, the genetic toolkit inherited from a common ancestor that lived somewhere between 3.5 and nearly 6 million years ago. The goal was straightforward: confirm exactly how and when these 5 predators split apart, and finally settle the Panthera branch of the tree the same way the 2006 study had settled the rest of the family. Instead, the genomes contradicted each other depending on which piece of DNA researchers examined. One stretch of the genome suggested lions and leopards were each other's closest relatives. A different stretch pointed to jaguars and lions instead. A third region placed the snow leopard closer to the combined lion and leopard branch than any accepted classification had ever proposed. On a normal, cleanly branching family tree, this should not happen. Once 2 species split apart for good, their DNA is supposed to drift further apart over time, consistently across every part of the genome. Instead, researchers were looking at a genome that told several different, mutually exclusive versions of the same story depending on which chromosome they consulted. There were 2 possible explanations. The first, called incomplete lineage sorting, is a normal statistical quirk that occurs when a species splits rapidly into several new lineages within a short window of time before ancient genetic variation has fully sorted itself out, producing a scrambled signal without any actual interbreeding involved. The second explanation was far stranger: that these 5 species, separated for millions of years, had continued mating with each other long after they had already become distinct species, repeatedly exchanging genetic material across a boundary that should have been closed for good. To tell the two apart, researchers ran statistical tests, commonly known as D-statistics, that measure whether shared genetic variants between two species are distributed the way random chance and incomplete lineage sorting alone would produce, or whether they cluster in patterns that only direct interbreeding between two specific lineages could explain. Running that test requires an outside reference point, an animal distant enough from all five Panthera species to serve as a neutral baseline, and researchers use the domestic cat genome for exactly that role. The logic works something like checking a family's finances for signs of a secret loan. A few small, scattered similarities between two accounts could be coincidence. A large, concentrated block of matching transactions appearing in specific places and nowhere else means money actually changed hands. When researchers ran the numbers on Panthera, using the housecat genome as the fixed outside reference, they found the second pattern, not the first. This was not noise left over from a fast radiation. Genuine, historical hybridization had left its signature across the genus, again and again, long after these animals had already become separate species by every standard definition biologists use. The clearest case involved lions and leopards. Despite roughly 3.5 million years of separation, the genomic data showed clear evidence that lion and leopard lineages had interbred at some point in their shared history, leaving detectable leopard genetic material inside modern lion genomes. The lion lineage in particular carried the widest signature of ancient admixture of all five species, likely a byproduct of its historically enormous range, which once stretched across Africa, the Middle East, and deep into Asia, overlapping with more relatives than any other big cat. That range is exactly what brings the opening snow leopard back into the picture. Researchers found that the snow leopard lineage also carries a detectable trace of ancestry from that same combined lion and leopard ancestor, a lineage it has no business sharing genetic material with, given that snow leopards specialize in terrain almost no other big cat can survive in. The crossing points to a period sometime in the Pleistocene when the ranges of these lineages briefly overlapped, likely at the shifting edges of Central Asia, before separating again for good. That mattered for a separate reason. The same study identified genes under strong species-specific natural selection in each of the five cats, including the EGLN1 gene identified back in the original 2013 comparison, the one behind the snow leopard's own independently evolved solution for surviving low oxygen at extreme altitude, alongside distinct selection signatures in the jaguar's skull and jaw and in the tiger's reproductive biology. Researchers also identified specific genes showing signs of positive selection, working in tandem with signals of historical introgression, including one called ESRP1, tied to skull and facial development, and another called SSTR4, involved in hormone signaling, both flagged in regions where ancestry from a different Panthera lineage had clearly moved in. Researchers suspect that during the extreme climate swings of the Pleistocene, when big cat populations across Asia and Africa periodically collapsed to a small fraction of their former numbers, occasional interbreeding with a neighboring species may have injected badly needed genetic diversity into populations that might otherwise have been pushed toward extinction. In that light, the hybridization events look less like an accident of proximity and more like a survival strategy written into the genome itself, one nobody had thought to look for until the whole genus had finally been sequenced side by side. Subscribe to not miss the next one. If the 2017 study had stood alone, it might have been treated as an interesting anomaly confined to one corner of the cat family. It was not. A follow-up study published in 2025 focused specifically on jaguars, lions, and leopards, went further, concluding that historical hybridization between these three species was so extensive that it actively overwhelms attempts to reconstruct a single, clean species tree using standard methods. The team drew on newer, higher quality chromosome level genome assemblies that had become available in the years since the original 2017 study, giving them a far more detailed view of exactly where in the genome the conflicting signals were coming from. Depending on which statistical model researchers applied and which stretch of the genome they analyzed, the calculated relationships between the three species shifted, sometimes placing the lion closest to the leopard, sometimes closest to the jaguar, with no single answer holding up consistently across the whole genome. The paper's authors argued that a single bifurcating tree, one clean split after another, is the wrong shape for describing how these species actually came to exist. A tangled web, with genetic material crossing back and forth between branches long after those branches had technically become separate species, fits the data far better than a tree ever could. The same pattern, on a smaller and far more recent scale, shows up much closer to home. The Near Eastern wildcat that gave rise to every housecat on the planet has a close relative, the European wildcat, a separate subspecies that still survives in patches of forest across Scotland, France, and parts of Eastern Europe. As domestic cats spread across the continent alongside human settlements, they began interbreeding with these wild populations, and they have never fully stopped. Genetic surveys of wild living cats across Europe regularly turn up individuals carrying substantial amounts of domestic cat ancestry, to the point that conservationists now list hybridization with domestic cats among the greatest threats to the genetic survival of pure European wildcat populations. Unlike a shrinking habitat or a disappearing prey base, this threat cannot simply be fenced off. Every generation of interbreeding dilutes the wild gene pool a little further, and once enough domestic ancestry has entered a population, there is no genetic procedure that can remove it again. The same species boundary that supposedly separates a wild predator from a housecat curled up on a couch has been quietly blurred for thousands of years, the same way the boundaries between jaguars, lions, and leopards blurred millions of years earlier. What emerges from all of this is a picture of the cat family that looks nothing like the clean, orderly diagram most sources still print. Every living feline does still trace back to a single common ancestor roughly 11 million years ago, and the 8 major lineages identified in 2006 remain broadly accurate as a starting map. But the branches of that tree were never as sealed off from each other as the diagram suggests, and no amount of additional sequencing has made the picture any simpler. Species that split apart millions of years ago kept finding their way back to one another, whether that meant a snow leopard carrying a trace of ancestry from a lineage that never should have reached its mountains, a lion carrying leopard ancestry across an entire continent, or a European wildcat's bloodline steadily dissolving into the genome of the world's most common house pet. Every one of these findings depended on the same basic tool: DNA old enough or detailed enough to hold onto a record its owner never knew it was keeping. A snow leopard alive today has no way of knowing part of its genome traces back to a lineage it was never supposed to meet. A modern lion carries no memory of a leopard ancestor. And the animal sleeping on a couch somewhere, 95.6% identical in its DNA to an Amur tiger, despite 10.8 million years of separation, has no idea it is carrying around a genome that quietly rewrote what an entire family of predators is. The evidence sat there anyway, inside the genome itself, for millions of years, waiting for sequencing technology to finally catch up to it. For cats, a species boundary was never the sealed wall it was assumed to be. It was closer to a fence with a gate that kept swinging open for millions of years, letting through exactly what was never supposed to cross.